So hello everybody. Today it's my pleasure to introduce Rosa Santomartino from the University of Edinburgh. She is a Leverhulme Research Fellow there at this institution, and her talk will be about microbial interaction with terrestrial and extraterrestrial rocks on the ISS. Welcome, and the floor is yours. Thanks a lot.
So yes, I am a research fellow from the University of Edinburgh, and more specifically for the UK Centre for Astrobiology here. And today I'm going to talk a little bit about what we learned from the point of view of microbial interaction with terrestrial and extraterrestrial rocks by two studies that we performed on board the International Space Station, BioRock and BioAsteroid. So let me check if I can go on. Sorry, of course, the thing is doesn't allow to go on, so a little patient. Maybe like that. Right, okay. Now, what we are going to talk about today? Well, first I'm going to just briefly summarize the roles that microorganisms perform for us on Earth in our everyday life, then possible roles of microorganisms beyond Earth. And I'm going to focus more on biomining, particularly space biomining. And then I'm going to focus on the two space experiments that I mentioned before, so BioRock and BioAsteroid. And finally, we will conclude with like a summary, some conclusion, and future perspectives from what we learned. Now, I'm sure I don't have to convince you about the fact that microorganisms are quite important, and I also probably you don't need to. I don't need to explain to you how important they are for our own survival here on Earth. But just to summarize some of the roles that microorganisms perform in our everyday life here on Earth. Here I just want to summarize some of them. So for instance, as you know, in early Earth, microorganisms started producing at some point a large quantity of oxygen because they started performing photosynthesis, and this is called the Great Oxidation event. And basically we have to thank microorganisms for the fact that for the oxygen concentration that we have in our current atmosphere. For soil, well, microbes are essential of course, for soil formation and for the soil ecology because of their involvement in carbon, nitrogen, sulfur cycles, and also because of the fact that they enrich the sterile soil with the organic compounds so that soils become actually fertile for our crops. But also getting more into the human side of it, a lot of biotechnologies and bioindustries involves microorganisms to work. For instance, microorganisms are essential in the food industry, but also in the energetic industries. This is just one example indicating biofuel, which is formed sometimes. There are a lot of research by understanding how microorganisms can help the formation of biofuel, but also in the production of polymers, like for instance biocomposite or biowool, cotton, and so on. Very importantly, in the drug industry, microorganisms are basically becoming essential. Just to mention one drug which is made nowadays all thanks to microorganisms: insulin and antibiotics, of course. But also from an environmental point of view, microorganisms are actually essential for the recycling of waste. And I'm not only talking about organic waste, but also plastic waste, for instance, as well as electronic waste. And again, from another perspective, which is related to an environmental issue, microorganisms are used in a technology called bioremediation, which helps removal of toxic pollutant from polluted environments. And last but not least, also in the process of biomining, which I'm going to talk more in a couple of minutes. Now, these last four applications here, they are quite diverse one to each other, but they rely on similar biochemical reactions. And these biochemical reactions are the reactions of bioleaching or biomining. Now, when normally these two terms are used as synonyms synonyms, one of each other, but in the idea that I got is that actually I talk about bioleaching more when I'm talking about the chemical reaction, and I'm talking about biomining more in the when I'm talking about the industrial, from the industrial point of view. However, what is biomining? It is the process of using microorganisms to extract elements and metals of economic interest from rock or mine waste. And as you can see here, they also say that these techniques are also used for bioremediations, basically. I'm not going to get into the details of the chemical reaction, but I would just want to mention that the chemical reaction are quite diverse depending on the microorganisms that is used. The image that you can see here is the image that comes from chemolithotrophic microorganisms, but it is also known that heterotrophic and organotrophic microorganisms can perform biomining as well, or bioleaching if you want. However, what I want to point out here is the fact that nowadays on Earth, 20–25% of copper, around 5% of gold, but also other elements like uranium, zinc, cobalt, rare earth elements, and so on, are currently extracted on Earth thanks to the biomining technique. And there are some advantages in using biomining in respect to traditional mining. For instance, it is generally considered more complementary. It's considered complementary in respect to traditional mining because it helps improving the rates of metal recovery from mine waste, for instance, but it is also generally considered more economic and also more environmental friendly. Now, we've seen how many roles and tasks microorganisms perform on Earth, and also things that are strictly related to our own survival and thriving here on Earth. You can imagine how important they could become on an extraterrestrial environment beyond Earth, in other terms. For instance, some of the possible roles that they could perform for us on an extraterrestrial settlement, for instance, included the generation of oxygen. We've seen that they were quite important in early Earth, so we think that bacteria, microorganisms, actually, together with plants, can really help us on an extraterrestrial settlement by generating oxygen. Generation of food, we've seen that they are quite important in our human food industry. So it is believed that they could become quite essential on an extraterrestrial planet, and many studies are trying to understand which microbes can be better used to produce food available for astronauts or cosmonauts, if you want. And related to this, there is also the production of soil out of extraterrestrial rocks or regolith. This is also related to the generation of food, if you want. However, microorganisms can not only bioremediate, I would say, regolith, extraterrestrial regolith, by removing toxic compound, but they can also enrich it by organic material thanks to their action, basically. Then this is something that I'm getting more and more interested in, the recycling of waste. Now, as you know, the space station is an enclosed environment, and probably the first settlements are going to be closed environment as well, because the environment outside is not going to be very suitable for life. So in an enclosed environment, it is necessary to recycle as much as possible because the resources are going to be limited. So microorganisms can really help for this perspective as well. And last but not least, they can also help in space bioindustries. For instance, the production of drugs in space, but also biomining and many others. Now, if you notice, all these possible roles of microorganisms in space have all the final aim to basically sustain what we call life support systems. And when I talk about life support systems, I always like to mention the Melissa project, because this is a project that I love. I think it's very fascinating, and it's been on for, I think, more than 30 years now. What is this? It's basically just a loop of interconnected compartments. Most of them includes microorganisms, but also plants and humans. And the concept is that any compartment is going to produce some waste material that is going to feed the next one, and so on, so that you get this kind of virtuous loop in which this is is going to be kind of self-sustaining. But related to the concept of using microbes for life support systems, there is also the concept of in situ resource utilization. What is that? Well, the majority of space agencies agree now that the future for human space exploration in the next 10 plus years is to move farther and farther from Earth, and trying to establish a settlement, a stable settlement on the moon first, and then moving toward Mars. So in this context, we cannot think on relying on a constant resupply of resources from Earth, and we have to think on actually relying on resources in the place in which we are going to settle, particularly if we are talking about another planetary body or something like that. So this is the concept of in situ resource utilization. And from this perspective, the concept of the possibility of performing biomining in space is rising and increasing interest. Well, it's probably easy to understand why, because the idea of using microorganisms to extract elements, useful elements, maybe for constructions or structures and so on, directly in the place in which we are going to settle is quite intriguing and important from the concept of in situ resource utilization. And when thinking about the possibility of performing biomining in space, we normally think we are interested in understanding if this is feasible on the Moon and Mars, of course, because of what we said before on these are the next goals for human space exploration, but also some asteroids for several reasons. One of these is the fact that some asteroids are rich in precious and rich elements, like for instance, platinum group elements. However, the conditions that are present in space are, of course, going to be different in space, and on these planetary bodies are, of course, different on respect on those on Earth. And one condition which is different is, of course, the regolith composition, the rock composition. But another condition that we need to consider to understand if space biomining is feasible is the gravity condition, because all the planetary bodies that we have listed here have a gravity which is lower in respect to the terrestrial one. We have lunar gravity, which is 0.16 g, Martian gravity around 0.4 g, and on asteroid we have often microgravity, centigravity for the larger asteroids, like for instance Ceres or Vesta. So when we think about performing biomining in space, we first need to understand if gravity somehow influences the way in which microbes interact with the mineral surfaces. And with this idea in mind, we performed the two first biomining experiments on board the space station. First is BioRock, which was launched in space in 2019, and the second one is BioAsteroid, which was launched later in 2020-2021. So let's focus on BioRock first. BioRock was first proposed in 2008 by Professor Coquelle here at the University of Edinburgh, and it was proposed in the frame of the ESA Geobiology for Space Exploration and Settlement topical team. But it was launched in space in 2019, so as you saw, it took quite a long time, 11 years to actually get to reality. And yes, it was proposed by Charles Coquelle, and as you can see here, a lot of big teams of people, different people, different groups were absolutely necessary for make this a reality. And here I want to show you a video which I always like to show when I talk about BioRock. It was prepared by NASA in preparation for the experiment. So when I show this video, I always have to mention that you will hear them speaking in the future tense, but that's because it was prepared before the launch. Now, of course, it's long gone and long finished, but I still want to show you this video because I think it explains very well the rationale behind BioRock. So let's hope that the audio works fine.
The International Space Station will soon host some of the smallest miners in the universe, microbes. Microbes growing on the surface of rocks can gradually break them down and extract useful minerals and metals. This is a process called biomining. As we explore space, we are seeking to use biomining to turn rock and regolith into soil for growing plants and food. But before we can use this technique in planetary settlements, we first need to test it in space. On the space station, bioreactors will be placed inside a centrifuge where microbes will grow on rocks in microgravity and simulated Martian gravity. Investigators will examine how 3 types of microbes behave within pieces of basalt and evaluate how well the different microbes extract elements from the rocks. The findings will be compared to ground-based results. We hope to gain insights into how microbes interact with rocks in microgravity and how we might use them in our exploration of deep space.
Subscribe for more.
Oh, not again. Okay. Okay, so I haven't received any message, so hopefully you heard it right. So let's get a little bit into the details of BioRock. We first needed to select the organisms, the right microorganisms for this experiment. And the right microorganisms, we thought, needed to have some characteristics in common together. For instance, it needed to either come from natural rock or soil environments, because this would have been a good indication that it could survive and thrive actually in the presence of rocks. And also, or it could he needed to have a demonstrated capacity to extract elements from the rock. So in other words, to perform bioleaching or biomining. They also needed to be desiccation resistant because of the way they have been launched in space. I'm going through this later. And also, if possible, they needed to be previously used in space experiments, because this would have been an indication that they could, again, thrive under space conditions. They needed to be easily studied in the laboratory for, of course, to have to enhance the majority, to enhance the possibilities of having more and more data out of this experiment. And last but not least, it needed to be safe for the crew. So eventually we selected these 3 different microorganisms. They are all bacterial species. The first is Swingomonas dissecabilis, which was provided by our group here in Edinburgh. Second, we have Bacillus subtilis, which is one of the most widely used microorganisms in space microbiology, and was provided by DLR, German Aerospace Center, and they are quite the experts in these organisms. And also Cupriavidus metallidurans, which also was provided by SIKSN in Belgium, and these people here are also the experts on this microorganism. And also these 2 had demonstrated capacity to extract elements from the rocks. We also needed to select the rock substrate, and we selected basalt for 2 main reasons. The first is that basalt is quite a porous rock, so it provided a good support for the bacterial colonization. And the second is that basalt is found on both Moon and Mars, meaning that it provided also a good analog for these 2 planetary bodies. We selected a rock specimen from Iceland, and we cut it into basalt-like pieces. Here you can see an example of it. They were roughly 15 millimeters per each side and 2-3 millimeters thick. They all look like that. And then we prepared the samples. So basically we inoculated each basalt slide with a different bacterial species. Each bacterial species had a different protocol for sample preparation, because it was basically designed to improve the survival and the thriving of each single species. So every basalt slide was inoculated with a single bacterial species, but we also had non-biological control, which was just sterile basalt slide. And then we have inserted the basalt slide into the hardware that was, the hardware is called Biomining Reactor, or BMR, and has been produced thanks to Kaiser Italia. It was specifically designed for this experiment, and as you can see here, each hardware can allocate to different basal slides. So basically each hardware allocated to different samples. This is again the hardware which was inserted into the external container. So basically what you see here are example of hardwares ready for the launch on board the ISS. And then the experiments, so the samples have been launched on board the International Space Station in July 2019 from NASA Kennedy in Florida, thanks to the SpaceX Commercial Resupply Service 18. Here are just some images. This is me on the launch site, and this is the launch. Here is when the Dragon capsule arrived on the ISS, and here you can see the astronaut Luca Parmitano, which is installing the hardware inside the cubic incubator. And I just want to mention there was they showed this incubator also in the video that I showed you before. Basically, you can see that some hardwares have been, so here you can see a round shape. This is a centrifuge, which allowed the simulation of terrestrial and Martian gravity. I'm coming back to this in a second. And then there are also spaces allocated outside the centrifuge, and these samples were basically just subjected to microgravity. In fact, these are the conditions that they were subjected in space. Basically, we prepared 4 different set of samples, 4 identical set of samples, I should say, each one containing 3 replicates of each bacterial species plus the non-biology controls. 3 sets of samples have been launched in space, and they have been subjected to either microgravity or simulated Martian gravity or simulated terrestrial gravity. So these 2 gravities here, these 2 simulated gravities were possible because of the centrifuge inside the cubic. And then a 4th set of samples stayed on the ground, and these were subjected, of course, just to real terrestrial gravity. So it was for us an extra control. The samples have been they grew in liquid culture in the presence of the basal light for 21 days at 20 degrees. And here are just some other images of the astronaut Luca Parmitano, which is taking some pictures of the hardwares. And basically, this photo acquisition was performed at the beginning and at the end of the experiments. At the beginning, just to check if the medium was injected sufficiently, successfully into the hardware, so that we knew that the experiment started, and at the end of the experiment, just to check if everything was okay, if the growth happened, and so on. Now, these images here are, of course, just qualitative images. They don't provide any quantitative data, but still, I want to show you the images that we got out of it, because although, again, being just qualitative data, it's still the photo session still reveals some interesting features that were present in space, but some of them were not visible anymore when we collected back the samples. So without these pictures, we wouldn't know about that. The first feature that we observed is the presence of these aggregates that are indicated here with the yellow arrows. You can see them particularly for Sphingomonas, Dessecabilis, and Cupriavitus metallidurans. This is probably some biofilm formation or something like that. And you can see that it was present at higher gravity, so Martian and terrestrial gravity, but not in microgravity. And the second features are the gas inclusions, or if you want a less fancy way, just bubbles. You can see that the gas inclusions indicated here with the blue arrows were quite more numerous and also smaller in size in microgravity in all the conditions tested in respect to higher gravities. And these particularly were not visible anymore, of course, when we collected back our samples on Earth. So again, without these pictures, we wouldn't know about this. After 1 month of being in space, the samples came back to us on Earth. And this is me and Professor Coquel just happy because we retrieved the samples. So let's get into the results. What did we get out of the BioRock experiments? The first thing that we've done was to analyze basically the final cell concentration so we wanted to check how well the microorganisms grew in these experiments. And to do that, we checked basically a portion of the liquid culture, and we analyze them by two methods, which are direct cell counting and optical density. And with this, we discover two main things. The first is that the difference that has been found between the that we found some differences between some of the ground controls and the space samples. For instance, you can see this difference here. In this case, we found that there was a statistically significant difference between the ground control and one of the terrestrial gravity, and we can also find it here for optical density. The interesting thing, though, was that the only statistically significant difference were found when, in few cases, but only when a ground control was compared to space samples. But we've never found any difference in final cell concentration between the space samples. And what does it mean to us? So the explanation, actually, what does it mean from the, let's say, a biotechnological perspective? Because of how the experiment was designed, we don't know what happens during the 21 days of growth. We only know what happens we only know the final cell concentration. So what happens at the 21st day of growth. So we don't know if the growth curve happened like that, if there was a longer or shorter log or lag phase, and so on. All we know is that after the 21st day, all bacterial culture reached the same final cell concentration regardless of the gravity condition. And this is a good indication that biotechnological application in planetary bodies with a lower gravity in respect to Earth will actually be possible, because gravity won't negatively affect the final cell concentration, so the bacterial growth. Ralph Muller also analyzed the spore formation of Bacillus subtilis. So among the 3 microorganisms that have been tested in space, only one was able to form spore, and this one is Bacillus subtilis. So he analyzed several stuff like the spore formation, the colony morphology, and the pigmentation, and he could find no differences in all in any gravity conditions. So gravity did not affect even these things. And then from the point of view of biomining, more strictly biomining, or if you want, bioleaching, we focus on 2 different types of elements here. Rare elements that have an enormous impact on our everyday life, because they are used in any electronic devices that we use, even the simplest one. So we thought that from a space perspective, in which we are going to rely on electronic devices even more than now, they are going to be relevant. And we also focused on vanadium because it has a strong industrial interest, since it is used in structural materials, generally to enhance the hardness and the resistance of the structural materials. So considering the harsh conditions that are present in space, we thought that these 2 types of elements will be interesting from space biomining perspective. And we found that Sphingomonas dissecabilis was actually able to biomine rare earth element in all the gravity conditions that we tested, particularly in all those tested in space. And we also found that 2 microorganisms out of 3, Sphingomonas dissecabilis and Bacillus subtilis, were able to biomine vanadium in all the gravity conditions that we tested. So this is a very good news from space biomining perspective, because it is first indication that in principle, space biomining is going to be achievable under a range of gravity conditions. We also tested biofilm formation. And the way we've done that is we basically we've taken the basal slide and we observed them, the top layers of the basal slide, under confocal scanner, laser scanner microscopy, by staining each basal slide with cyber gold, which is a staining specifically for nucleic acid. So what you see here in green is basically the biofilm formation. And what you can notice is that both Sphingomonas dissecabilis and also Cuprigravidus metallidurans formed biofilm on the rock. Bacillus did not, but we have other data that I don't have. I cannot show you today because I have finishing analyzing them, but we know that they grew very well on other type of surfaces, and also remember that it's still was able to extract vanadium, so apparently biofilm as a first indication is not necessary for Bacillus to extract elements. However, Sphingomonas dissecabilis and Cubiavidus metallidurans were able to form biofilm on the rock surfaces. And as you can see here, by when we quantify the fluorescence of that we observe here, we can see that there is a trend for an increase in biofilm formation at increasing gravity regimens. I have to talk about trend because, as you can see here from the high variability that you can see here, these differences here are not statistically significant, but still you can see a trend of increase for biofilm formation, and you can also appreciate it hopefully from the images. You can see here like less green and then fluorescence increasing with the higher gravity regimens. So from BioRock, basically, we discovered that low gravity is likely not going to be a limit for biotechnological applications in space. We also demonstrated for the first time biomining on a space station, and we also demonstrated biofilm formation on rocks in space. Now, after BioRock, we performed the second biomining experiment in space, which we called BioAsteroid. It is a little more recent, 2020-2021. And the rationale is quite similar to BioRock, but there are some key differences. For instance, in this case, we still use the bacterium Sphingomonas dissecabilis, so a bacterial species still, but we also enlarged, let's say, our knowledge on microbial functioning related to space biomining by using a fungus, so another type of microorganisms, and particularly we use Penicillium simplicissimo, and also a mix of the two, because we wanted to understand if like a small consortium could enhance or rather hinder the biomining capacity of these two microorganisms. And also we changed the rock substrate. So for BioRock, we used terrestrial basalt. For this experiment, we decided to go for real extraterrestrial material, and we actually used a crushed elchondrite. So not only we used an extraterrestrial rock, but also we changed the structure of the rock. Not the structure of the rock. We crushed it. While for BioRock, we were also interested in checking the biofilm formation on the rock. This is why we went for basalt slide. In this case, we decided to crush it because biomining experiments and technologies usually crush the rocks in order to increase the surface area of this. So this is what we've done here, and hopefully you can appreciate it from this picture in which you can see the beads, little pieces of the elchondrite that's been crushed. Also, the experiment is slightly smaller in respect to BioRock. We had a set of samples that had been launched on the International Space Station and were subjected to microgravity. And then we had another set of samples that stayed on Earth and were subjected to real terrestrial gravity. Then the rationale was quite similar. So liquid culture were grown in space or on the ground in the presence of the rock, the meteorite rock pieces for 19 days at 20 degrees. And what about the results? Because the experiment is more recent, unfortunately, I don't have too many results to show you at the moment, but I still want to show you some preliminary results if you want. The first is some scanning electron microscopy images. Basically, when samples came back from space, I've taken the rock pieces and I treated them, coated them with gold, and I analyzed them under scanning electron microscopy to check for some indications of rock weathering or microbial growth on the rock pieces. And when I observed the samples that were grown in the presence of the bacteria, Sphingomonas dissecabilis, we found these beautiful structures here that you can see here even more enlarged that really resembles cell structures, as you can see here. So we believe that this is some indication of biofilm formation on the meteorite rocks. However, we still have to double check this, because when we check non-biology samples, we also found some structure that are not exactly the same as the one for the bacterial samples, but still quite similar. So we are now keep on analyzing this to double check if these are real bacteria. And the first indication seems to be consistent with the fact that this is actually biofilm. But more interestingly, when we checked samples that have been grown in the presence of the fungus, so the only fungus samples and also the mixed samples with bacteria plus fungi, we found that in both cases the mycelium was successfully growing in on the rocks in space. So this is, in my opinion, quite nice images. Then, from the point of view of biomining, again, the data are still ongoing. I'm still analyzing the data, so I'm sorry I don't have a graph to show you here, but I wanted to give you some preliminary data, some preliminary indication of what we are finding while analyzing the samples. And we found that some elements have been extracted in a higher extent in space, while some other has been extracted in a higher extent on Earth. And I want to mention something here. For instance, platinum looks like it's being extracted in space at a higher extent in respect to the ground, which is quite interesting. So basically, we have three things to point out here. The first is that with this we have a first evidence for extraction of elements, like for instance rich elements like the platinum group element from the meteoritic material in space. Also, you can notice that there is a larger number of elements that have been extracted in space in microgravity, therefore, than compared to terrestrial gravity. And also the gravity seems to have a variable effect on biomining of different elements, because you can see that the elements that have been extracted here are different than those that are being extracted here. Although, again, these are just preliminary data. And then also the consortium partially changed the pattern of extraction, because yes, some elements, for instance, scandium here has been extracted both in the consortium and the fungus, and same for the platinum, but some other elements have only been extracted in the consortium and vice versa. And this is the same here. So, to conclude, basically, with BioRock and BioAsteroid, we provided the first demonstration of biomining on a space station, particularly from meteorite rock with BioAsteroid. We also demonstrated space biomining using organotrophic microorganisms like bacteria and fungi, because, as I mentioned before, the majority of time when biomining or bioleaching is studied by using chemolithotrophic microorganisms, but actually organotrophic microorganisms can also be used, and we use this in this case. Also, we demonstrated that space biomining is in principle achievable under a range of gravity regimens, and that also final cell numbers seems to be not affected by gravity conditions in a few weeks, which is a good news from the point of view of space bioindustrial applications. We also discovered a novel biomining microorganism, which is Phingomonas dissecabilis. It was not known as a biomining microorganism before. And also we provided the first demonstration of microbial interaction with meteorite material in space, in microgravity. Now, there is a lot that we still need to understand. For instance, we need to get more insight on the effect of gravity and spaceflight on microbial processes, and this is a general concept, I think. We need to select the best microorganism for a given application, because we cannot give for granted that some application that is available on Earth, is going to be immediately applicable to space. We first need to test it. And of course, from the point of view of biomining, we also need to scale up the system a little bit and develop the technologies, because yes, we found that gravity in our experiment didn't have an effect on biomining, but also on bacterial growth. But our hardware was quite small. It was only 5 ml of volume. So in larger bioreactor, this could change, and this needs to be addressed as a question. Another thing is that understanding if we could have the biomining compartment of biological life support system. Now, biomining is not normally considered a biological life support system, related to biological life support system, but we recently published a review paper in which we proposed that a biomining or bioleaching reactor can not really add it to a system, a life support system like Melissa, but at least enhance and support some ways the life support system and vice versa, because they could produce useful stuff and vice versa. And last but not least, we also need to learn how to keep all the process sustainable. And this is something that I get more and more interested. In fact, I recently started a new project in which I'm going to start plastic biodegrading microorganisms for terrestrial, but particularly space applications. It is funded by the Leverhulme Trust, and I'm working on this here at the University of Edinburgh. If you want to know more, feel free to contact me. I'm very happy to collaborate. Now I'm concluding my my talk by thanking, of course, all the amazing BioRock and BioAsteroid teams, which, as you can see here, is full of a lot of people. And I would like to thank you for your attention, and I will be happy to take your questions.
If I can have the first question.
Yes, please, Petra.
Yeah, thank you very much, Rosa. That was a great overview. And also, after some time after BioRock, there seem to be some more results I did not see before. So I have a question concerning the comparison between BioRock and BioAsteroid. Do you have an idea about the surface areas of the porous basalt in the discs used for BioRock and the surface area of the crushed chondrite material? As it was obviously much higher.
Yeah, so for BioRock, we didn't do that, and it's actually sorry, I'm having. Oh, okay, sorry, I was reading the chat. Yes, with BioRock, we didn't do that. We know that it's quite porous. I wonder, now that you mentioned that, it's actually a good idea to check if we can analyze them, because we still have some samples. So it's actually good suggestions for BioRock, and you've seen that I have new data from biofilm formation, and I'm working on that paper, so that could be something that I will write down to analyze. While for BioAsteroid, we are also analyzing the surface area, so that we can, when I will have more polished data, hopefully I'm going to be aware of the surface area that was available to the microorganisms for the extraction. So thanks for the suggestion. I'm going to write this down.
Yeah. And what I also think it's interesting that, as you said in your presentation about BioRock, that some microbes like Bacillus subtilis are effective in biomining, at least for certain elements, but they do not form biofilms on top of the basaltic slide.
Yeah, that's really interesting, and probably, well, you know about biofilm formation, you know about Bacillus subtilis. So I wonder if there is any particular biomining mechanisms by which they can extract elements without forming biofilm. So biofilm helps biomining and bioleaching in general, but there is not. There are many mechanisms for that. In literature, you can find that there are like contact and non-contact mechanisms for bioleaching, although eventually the chemical reaction, they are the same, but the general idea is that contact mechanisms are more related to biofilm formation and the others are not, or at least are not strictly related. That there is still some involvement. So I wonder if there is any indication on the way by which Bacillus can extract, like, you know, the mechanisms.
So I am not aware of detailed concepts about this. So I do not know.
Okay, but maybe something worthy to study. I don't know.
Yes, of course.
Thank you.
Okay, there is also a question by Adrian Kish. What surprises did you have trying to do microbiology in space?
Well, many, I would say. Thanks, Adrian, first of all. Trying to do microbiology. Well, I guess, well, many things. If I get to more into the scientific point of view, is the fact that when you work on a bench, on a laboratory on bench, of course you have some limitation related to the grant, the money that you have. So it's not like you can do any experiment that you want sometimes. you still have limitation. But when you have to do a space experiment, you have to work with the engineerings and with, I don't know, in my case, I needed to understand, with several expertise of several different groups of people. So sometimes you want to do a lot of stuff, and then the engineering comes to you and tell you, yeah, but actually you cannot have like a 100 ml bioreactor. You have to compromise everything a little bit. So trying and understand how to have the best science possible with the limitation that you have. But still, maybe I'm making this sound like a bad thing, but it's actually something that it really inspired me, understanding how much there is behind the space experiment, not just, I don't know, also the fact that you cannot repeat the experiment. That was quite scary in some ways, because if you have a problem, you cannot easily do that again. But yeah, that's, and then of course it was extremely beautiful to do that. It was extremely beautiful to be on the rocket launch site. Yeah.
Okay, Chaitanya, please.
Right, thanks. Thank you, Rosa, first of all, for a very fascinating talk. That's some great amount of work.
Thank you.
I was particularly intrigued by the last set, which you discussed about the consortia versus the individual ones. And do you see of ways of constructing optimal consortia so that one can synergize the microbial communities in the best way possible to get maximum extraction in that way? Or are there studies on that, especially in this particular topic about that? And the second part was, now that, like, I mean, the evolutionary process of these microbes is quite fast, especially now if you're changing ecologies. So are there any concerns about if they might deviate from the designed goal over time? And what are your thoughts about that?
So from the first question, which was related to the consortia, at the moment I'm not studying that more in detail myself. Some people are studying something similar here at the university in my group, but they are studying that from a different perspective. Like, I know that there is people studying a natural consortia. So I call it consortia, but it's just 2 microorganisms, you know. Some people have isolated some consortia from an anaerobic pond, and they are using it to try and understand how this consortia behaves with some extraterrestrial material. I don't know the results out of it, but I know that they are working on that. From the biomining perspective, there are a lot of work related to biomining and consortia. So it is known that consortia are quite good from this point of view. I'm not aware of other type of works on this space related, but I'm pretty sure that there is people going to do that, because it's quite a clever thing to do. So I will be surprised if nobody else is working on that. And also from my perspective, it will be quite interesting related to space, because you know, the rocks that are present on Moon or Mars, or even if you want to mine asteroids, are quite diverse than those on Earth. So maybe one consortia or one microorganism that is working very, very well on Earth is not going to work as well on Moon or Mars or something like that. So and you also have to consider some other conditions, like I don't know, the perchlorate on Mars, for instance, or the lack of carbon sources and organic material, etc. So if you create a consortia in which you have, for instance, like a cyanobacteria that can produce the organic material, and then an organotrophic microorganisms, that this can really help from this point of view. So I don't know if I answered your question, because I did more questions, I think. The second thing that you asked me was, can you repeat the second? Sorry.
So the first part you definitely answered. I was exactly concerned about, like, how can one construct the synergies between them? Because if you finally want one microbe to actually do the work, it needs a lot of other things which maybe others can provide. The second part was about, once you have such kind of a consortia where you have these designed interactions which are set, but now we change their ecology, because now you put them on Moon or on Mars.
Right.
How do we control that they don't evolve to something different?
I think this is real for any biotechnological application that you want to do in space. So this is another thing that I think he needs to be addressed. So there are a lot of studies related to microbial response to radiations, for instance, or also growth on analogs of extraterrestrial regolith, and I think they are quite important. And at some point, so I guess like the data that I showed you today are okay, the first data on this, nice, but there is a lot that we still need to understand from this point of view, and because we really, I don't think we can ever predict any possible thing that could happen, and particularly if we go and establish a permanent presence in space, then nobody knows what happens when you have a permanent presence of microorganisms, let's say a biomining reactor on the moon, for instance. Nobody knows that. Needs to be studied. Definitely. I don't have any indication on my samples, I'm afraid.
No, thanks a lot. Yeah.
So may I comment on this?
Yeah, sure.
So I think for a permanent station on Moon or Mars, and for very long-term cultures used for biomining or for any other purposes you already mentioned at the very beginning, which could be done in space, there will be a kind of there will be mutations in the microbes, and there will be different adaptation processes, and therefore I think we have to make sure, if we really go in this direction, to plan and have stored new starting cultures. So if there is a habitat on the moon, then we can start again if something goes really wrong. But this will not be from, except for technical failures, this will not be from one day to the other, but this will be a gradual process.
Right, yeah, that's true, yeah. Yeah, and maybe, yeah, no, I agree. I think that this could be a solution, or we can, I don't know if we can predict it, you know, like we just have to keep in advance and have like a B strategy or something like that. That's something that I think this is why it's so important to study these things, because we need to know more and more on this.
Yeah.
Right. Thanks a lot. Thanks a lot, Peter, as well.
Thank you. Yeah.
Okay, are there any more questions now? I do not see any more questions. So I thank you again, and I would invite you for the next seminar, which is unsurprisingly in 2 weeks' time, with Klara Anna-Chapova from the European Space Agency. It will be a more political and societal theme, as I expect. Many thanks for today. Many thanks, Rosa, for coming and for giving us this nice overview. And see you then in 2 weeks. Thanks a lot.